The power of tiny clues. Molecular techniques in pathology. Think of a detective at a crime scene. In pathology, doctors are like detectives. Molecular techniques are their magnifying glasses. They look for clues at the smallest level molecules. By examining DNA and RNA, we identify disease causes. These techniques lead to better treatments. Molecular techniques are essential in pathology. DNA and RNA are like instruction manuals for our bodies. They tell our cells what to do. DNA is like the master blueprint. It contains all the information that makes us unique. RNA is like a messenger. It carries out the instructions from DNA. Each DNA molecule is made up of a unique sequence of building blocks called nucleotides. These nucleotides are like letters in the alphabet. They can be arranged in different combinations to create different genes. Genes determine our traits like hair color and eye color. RNA is similar to DNA, but it's single-stranded. It plays a crucial role in protein synthesis, carrying genetic information from DNA to the ribosomes. By looking at specific sequences in DNA and RNA, we can identify tissues and infective agents. This helps us diagnose diseases and develop targeted therapies. Imagine you have a single page from a rare book. You want to share it with the world. This is where PCR comes in. It's like a molecular copy machine. PCR stands for polymerase chain reaction. It makes millions of copies of a specific DNA segment. PCR has revolutionized molecular biology. It's used in genetic testing, forensic science, and disease diagnosis. Section 4. Unraveling the steps of PCR. PCR might seem complicated, but it involves a few basic steps. It's like a recipe with specific ingredients and instructions. The first step is denaturation. We heat the DNA sample. This causes the two strands of the DNA double helix to separate. Next comes annealing. We add primers to the mixture. Primers are short DNA sequences. They bind to specific regions of the target DNA. This marks the starting point for DNA synthesis. The third step is extension. We use an enzyme called DNA polymerase. It adds nucleotides to the primers. This creates new strands of DNA, complementary to the original template. These three steps are repeated many times. This results in an exponential amplification of the target DNA sequence. Section 5. Beyond PCR, other amplification techniques. PCR is not the only amplification technique available. There are other methods, each with its own strengths and limitations. For example, Lagasse chain reaction, or LCR, is another technique. It amplifies DNA, but it uses a different enzyme called Lagasse. NASBA, or nucleic acid sequence-based amplification, is another method. It amplifies RNA instead of DNA. This is useful for studying RNA viruses. SDA, or strand displacement amplification, is another technique. It uses a unique isothermal amplification mechanism. This means it doesn't require repeated heating and cooling cycles like PCR. These alternative techniques provide additional options for researchers and clinicians. They offer flexibility in studying different types of nucleic acids. Section 6. Real-Time PCR, a powerful advancement. Imagine you could watch a cake bake in the oven. You wouldn't have to wait until it's done to know how it's doing. Real-time PCR is like that. It lets us monitor the amplification process as it happens. Real-time PCR is an advanced form of PCR. It uses fluorescent dyes or probes. They bind to the DNA. As the DNA amplifies, the fluorescence increases. This allows us to measure the amount of DNA in real time. Real-time PCR offers several advantages over traditional PCR. It's more sensitive, faster, and provides quantitative data. This makes it an invaluable tool for gene expression analysis, pathogen detection, and disease monitoring. Section 7. Applications across medicine, from diagnosis to treatment. Molecular techniques like PCR have revolutionized many fields. In pathology, they help diagnose diseases like cancer and infections. They're also used in prenatal diagnosis. This allows us to detect genetic disorders in unborn babies. In infectious diseases, molecular techniques identify pathogens. This helps doctors choose the right treatment. These techniques are also used to track the spread of outbreaks. Molecular techniques are also essential in drug discovery and development. They help us understand how drugs interact with our bodies. This leads to safer and more effective treatments. From diagnosing diseases to developing new therapies, molecular techniques are transforming medicine. They offer a powerful toolkit for understanding and improving human health.